I wanted to thank the conference organizers, of course, for inviting us, and thank you for sitting through a couple of days of very intense discussions. It's been really outstanding to see the progress made in the field. I will be talking to you about tumor-infiltrating lymphocytes. It's a type of cell therapy for solid tumor. We really initiated our program regarding solid tumor, although we are now targeting heme malignancies, and I'll tell you a little bit about that as well today. We are a public company, so I draw your attention to forward-looking statements that I may make during my presentation. I know it's impossible to read it, so I'm just going to move forward. The technology comes from Steve Rosenberg's lab back at NCI. In 2011, he had published data on metastatic melanoma where he had shown responses in patients that had received back then standard of care with a 56% response rate, and what was quite impressive was a 24% CR rate in that patient population. After one-time treatment, many of these patients, in fact 19 out of 20 patients who were a CR, remained in CR at 7 to 10 years of follow-up. So this was an impressive piece of literature. So Iovance back then took a license to this technology in 2011. Around 2015 and 16, we started our clinical program. We had received an orphan drug designation for this product that is now called lifileucel for advanced melanoma. And in 2016, we decided that a long manufacturing process, which was what we had initially licensed from NCI, would be very difficult to be administered for patient population, which is quite late-line treatment experienced patients in today's landscape. They have all received anti-PD-1s, and if they have a BRAF mutation, they have received BRAF and MEK. So we embarked on shortening our manufacturing process, and we devised what we now call a Gen 2 manufacturing process, which is 22 days. And we started dosing patients because we were not completely confident that the manufacturing method changes has changed the clinical outcome. In 2017, we disclosed the data we had for this brand new manufacturing method in a cohort of study that is called Cohort 2 in our melanoma program. And the response rate was over 30%, which is quite appropriate for this patient population of highly relapsed refractory patient population. And therefore, we switched all of our studies into Generation 2 manufacturing method. In 2018, we proceeded to meet with FDA as part of an end of phase 2 meeting for our metastatic melanoma program, and the agency agreed that a single arm in our ongoing study is sufficient for registration for this particular product in advanced melanoma patients. And again, the patient population is quite late-line patient population. Currently, we have started the cohort, which is the pivotal cohort for this particular product. We started patient dosing in March of 2019, and we expect to complete enrollment in the first quarter 2020 and expect a BLA submission around late 2020. And we believe that this would be the first cell therapy product for solid tumor indications to come to the market. At the same time, around 2017, we started dosing patients in cervical cancer, and we then transitioned everybody to Gen 2 manufacturing method after we realized this is the better manufacturing method. And in 2019, we have released data from our cervical cancer patients. I'll discuss them in just a minute at ASCO, and we also met with FDA as part of an end of phase 2 meeting. As part of the interaction with the agency, we were awarded a breakthrough designation for Tyll product, which is again LN-145, for cervical cancer. And the agency agreed that the existing study could be supportive of registration if you proceed to define the patient population and complete enrollment. We believe that the current study in cervical cancer can complete enrollment in Q1 2020, and a potential BLA for that same product and same indication of cervical cancer is feasible by late 2020. So currently, the company is running 2 pivotal programs, one in metastatic melanoma and one in cervical. In addition to that, and we have an RMAT designation for metastatic melanoma and breakthrough designation for cervical cancer. In addition to that, we are now starting our work in hematologic malignancies. We are expecting to file an IND with the FDA by year-end in a new product that is called peripheral blood lymphocytes, or PBL, and we intend to initiate patient dosing in CLL in 2019 as well. So as key highlights in terms of the progress we have made in the past year or so, we have defined our registration path for lifileucel cell therapy for metastatic melanoma. We have defined our registration path also for cervical cancer with the agency. We have presented data for both of these indications at ASCO this year, where we showed that metastatic melanoma data is 38% response rate, and 2 patients, in fact, had reached a CR upon longer follow-up. And in cervical cervical cancer, we had 44% response rate. To put this in perspective as to what is available for these patient population, available care is really chemotherapy for both of them, and the response rate is about 4 to 10% currently for this patient population. In terms of what are the value drivers for the company, I wanted to focus on, of course, a large market opportunity. Solid tumor remains a market opportunity for cell therapies to come to the market. We have 4 company-sponsored studies in melanoma, in cervical cancer, head and neck, and non-small cell, as well as a basket study where we are bringing the combination of Tyll plus a checkpoint inhibitor to earlier line patients. We have an accelerated path defined for 2 indications, as I noted, and we continue enrolling patients into the pivotal program while we are looking into power of Tyll, which we believe is a platform in other indications such as head and neck and non-small cell. We believe that we have a scalable and proprietary manufacturing method. The method is a 22-day, as I noted. We have manufacturing providers in US and EU, and we have clinical sites in both US, EU, as well as Canada that are enrolling patients into our clinical program. Our manufacturing success rate is well over 90%, and that involves any patient that comes into the manufacturing facility, their tumor is collected to the time that the product is actually leaving the CMO. And we feel that the platform is quite broad, and in fact, we have started with academic collaborations in identifying proof of concept for new indications, and once a concept is well established, we bring that indication in-house to conduct company-sponsored studies. Just to kind of step back for a second as to what is Tyll. Tyll is the lymphocytes that come to the site of tumor and they initiate an attack on a tumor when it's detected in the body. Most of the times our immune system is quite capable and competent and is able to clear cancerous cells, which is why many of us don't have cancer at a young age. But in many cases, due to either genetic factors or environmental, cancer does develop and a tumor is in fact materialized. But the immune cells did come to the site of tumor and they tried to conduct the attack. There is a couple of reasons that they may not be successful. They may not be there in sufficient numbers, and there may also be because of the tumor microenvironment being quite hostile and blocking the effect of immune cells. But this was a discovery that Steve Rosenberg had, which was he recognized that these cells are at the site of tumor, and not only that they're there, but they recognize a neoantigen at the site of tumor. And so harvesting them has a benefit that you have a cell that has already recognized something about that tumor. So the cells are harvested, grown, and expanded and brought back to the patients. These are highly individualized therapies, so this is an autologous product. There is 100% of the patients we have shown have persisting Tyll product at day 42, and we presented this data at AACR this year. And we know that these cells have immunological memory, so they can circulate sometimes out to years. And this data was presented by Dr. Rosenberg, showing that Tyll products can be circulating out to about a year or so. And again, we stopped at about a year follow-up, but the cells are there. The remarkable data of deep CRs and durable ones were also noted in cervical cancer in Steve Rosenberg's studies as well, which is why we had selected cervical cancer as our second indication to pursue. At a high level, to compare CAR T therapy versus Tyll, the main difference is CAR T therapy is targeting a single surface tumor antigen. Specifically, the ones that are in the market are now targeting CD19, while Tylls are targeting multiple neoantigens, and we have a very polyclonal product. The second differentiator between CAR T therapy that is already in the market versus Tyll is the genetic modification. The 2 CAR T products that are in the market have genetic modifications, and the Tyll that we currently have in development does not have genetic modification. There's also differences, and this leads to difference in their safety profile as well, as we will disclose later. The manufacturing method that I sort of briefly touched upon is a 22-day manufacturing process. The tumor is excised at about 1 centimeter cube or so from the patient and is shipped directly to our CMOs. At the CMO, the tumor is fragmented, it's placed in media, and we allow these cells to depart the tumor and start the growth process. So the first 11 days of the process, both departure from the tumor as well as the growth is occurring, and the second 11 day of the process is a rapid expansion protocol where we rapidly expand the T cells into multiple billions, and by day 22, we harvest the cells, we wash them, and we cryopreserve them, and we are ready for infusion if the patient is ready. The patient receives a lymphodepletion regimen of cyclophosphamide and fludarabine, and if they don't have any other adverse events going on or anything else, they're ready to receive their TIL and be followed by up to 6 doses of IL-2. And this, the entire thing is the regimen is about 7 to 8 days duration from cyc-flu to completion of IL-2. In terms of intellectual property, we have a very broad IP landscape. We certainly cover both our manufacturing process, new indications, composition, as well as methods of treatment are heavily protected, and a few that have been granted are noted herein, but there's a much broader application process that has been taking place and claiming a number of different type of products, as I noted before. As part of a round of financing in 2018, we did commit to start our own manufacturing facility, and we have now selected City of Philadelphia as the location. We have a facility that is this is an artist rendering, of course. It's 136,000 square feet, and I'm told that this is the largest cell therapy manufacturing facility in U.S. That currently sort of in startup. The facility is expected to be ready around 2022, and we expect to launch our product through a CMO partnership and then switch to our own facility once it's ready. We expect, of course, a significant cost reduction once we are in our own facility, and we have committed to spend $75 million into the facility in addition to hundreds of additional jobs. I don't think I need to tell this audience about the potential market opportunity for solid tumor. I know that this has been subject to discussion through the course of the day today and yesterday, but the indications that I have listed on this slide are the indications that we are pursuing very actively in our development program, and melanoma and cervical in our pivotal stages, and the rest are in proof of concept or investigational stages. Our pipeline is noted herein. So the top two studies are the pivotal programs. The bottom two on the top of the table are the studies that are in newer indications that are being pursued. And a few of our investigator collaborative studies are noted in the bottom of the table. We have additional ones that we pursue, but two of these are, for example, through MD Anderson, as well as additional ones that are being pursued through collaboration. One is with Roswell Park that is underway. In terms of new products, we are focusing on heme malignancies as well at this point. We have a new process for a product that is called peripheral blood lymphocyte. The difference between PBL and CAR-Ts is that, again, this is a polyclonal product versus CAR-T, which is targeting a single neoantigen. So that's the difference. The same principle holds that we are still not genetically modifying these products, but CAR-Ts are genetically modified. And this particular product is coming from blood of CLL patients, for example. We start with 50 milliliters of blood, and it's a 9-day manufacturing process that we have developed. Our intention is to file an IND for this product by year-end and start patient dosing. In terms of our research directions, we certainly have a lot of broad interest in making TIL more potent. We have a broad platform, we believe, and we look at selected TILs, such as PD-1 selected products, which are expected to have better recognition of tumor because they have touched the tumor since they're PD-1 high. We are also looking at various other methods of changing the growth conditions for the TIL, including using different cytokines. And we certainly look at genetic modification, including transient genetic modification through RNAi technologies, as well as permanent genetic modifications through TALENT technology. So all of those are investigational approaches to the next generation of TIL products that we are investigating. In terms of our footprint, the company has around 130 employees. We are headquartered about an hour and a half flight north of here, about 15 minutes south of San Francisco in San Carlos. We have offices in Tampa, Florida, which is our research facility, in New York, and now in Philadelphia. And we have a presence in Switzerland as well, and manufacturing facility. The ground is broken in Philadelphia already. Our finances are shown here. I won't go into too much detail, but these are not certainly cheap therapies, and so we appreciate our investors' patience in supporting us in taking TIL into potential market. And in terms of what are upcoming milestones and what has been accomplished in 2019, we certainly started our pivotal program in cohort 4. We defined a registration path in cervical with FDA. Already we have presented our Gen 2 data in cervical and melanoma at ASCO, and we intend to additionally present 4 new directions as part of SITC. 3 of them, we have announced the titles. There are preclinical work, and there will be additional data that will be coming from our melanoma program with independent review committee data from our cohort 2 data. And filing a new IND and CLL program was one of our targets for 2019 as well. With that, I thank you for your time, and if you have any questions, I'm happy to answer it afterwards. Thank you.